Okay. Adi, before starting, I have few points for you. So hope you know we have already created WhatsApp discussion group. Yeah. So whatever discussion we have, we will be discussing here. Say for example, today if we have session, I will be posting the session link over there, and I will be mentioning the Indian time, exactly the what time the session starts.
Mm.
So please have a track over it. Okay.
Yeah, okay.
I also post this session. I will be creating a Google Classroom for you. So hope you have your own email ID,
Yeah, I
right?
have my own Gmail. I can
All
put it in.
right. I will be creating a classroom where we will be posting the session recordings, and also if there is any study materials need to be shared with you, we will be posting the link for you in the same classroom. Okay.
Oh,
So
okay.
I'm kindly requesting you to join the classroom using your Gmail account. Link will be shared in the group shortly. So whatever documents or if there is any link to be shared with you, everything will be posted over there. Okay.
Mm.
And
Okay.
also session recordings will be available for you and you can review it at your own pace, provided you watching it if you have any doubts. Okay.
Oh, okay. It makes
In
sense.
case if you have any other doubts apart from this, you can ask us in the next session. Or if you
Mm.
need any immediate clarification, you can also post us in the WhatsApp group. And also, I'm requesting you to give some time for Chandru to review it and respond. Okay. I
Oh,
hope
okay.
you can get it clarified by yourself at the maximum. If you cannot find any answer, you can definitely post it in the group. And whenever Dr. Chandru is available, he will try to respond to you. Okay.
Oh,
If
yeah.
not on the same day, it can be taken to the next session and he will clarify all your doubts.
Okay. Yes,
Okay.
makes sense.
This is
Mm.
regarding the technical part. And if you have any doubts regarding this, you can contact me in WhatsApp. Okay.
Oh, okay. Mm.
Fine. Good to start.
Yes.
Fine. Dr. Chandru, let's start. Yeah. Thank you so much.
Sir,
Doc-
you're on. Yeah.
Dr. Chandru, we cannot hear you. Sorry for that.
Yes. Am I audible now? Yes.
Yes,
Yes.
sir.
Yeah. So, Adi, have you found the brochure? Like, do you remember the brochure? What we shared?
No, sir. It's been a
No, you
long time.
for let me share it again for you. Actually, in the brochure, today's class will be about the basics of molecular biology. Fine.
Okay. Yeah.
So in your school time, hope you studied about the atoms, molecules. Am I right?
Yeah, yeah. Yes.
Correct. So one more small request. Like, can you please tilt the camera properly so that I can see your full face?
Good.
Yeah, it's going good. Good. Right. Got it, Adi? Like
Yeah.
today's class, we will be seeing about what?
Sir,
We'll see about, yes. So I am loading that. Just give me a minute. Okay. So meanwhile, let it get loaded. Meanwhile, what are the things you just gone through? Like, have you started preparing for this course?
mm, no, but like we have learned basics about like molecule, atom, compound in this class.
Okay. Right.
Mm.
No. So in the last class, if I remember properly, just you are shown about your previous research, correct?
Yeah.
You are shown about the previous research, and then I have suggested you like these are the basics will be required before you proceed. Yeah, before you proceed into your class, correct? Can you please
Yes.
recall the previous section?
No, sir. It's been like a long time. That's why.
A long time. Fine. Okay. Fine. Yeah. How about now? Is it visible? My screen is visible.
Yes, sir.
Yeah, thank you, Adi. So you just forget about all this right hand side. Just focus on the topics, right? So in the beginner session, we want to know about the protein structure. Like
Okay.
what is the principle of biomolecules? Like what are the different types of biomolecules are available, and what is a protein structure, right? And what is
Yeah.
structural informatics? Clear, right?
Mm.
Just a quick check with you, right? Do you know the number of amino acids present in the nature?
Um, there is 20 amino acid.
Very good. So there are more than 300 amino acids, but what you said the number is somewhat right, because the 20 amino acids refers to protein forming amino acids. Got my point?
Yeah. Okay.
And correct. The 20 amino acids are what? You can make a
Mm.
note of it. Like they are protein forming amino acids. Clear? And one more thing, there will be a recording. So you no need to write all the stuffs, but whatever you feel important, you can make a note of it. Fine,
Yeah.
Ali?
Okay. Yes.
Yeah. Yes. So why we not want to know about the amino acids, in the sense, by knowing amino acids, we can try to understand how these amino acids are getting combined. You
Okay.
got my
Yes.
okay. So what is the important thing by knowing the combinations of amino acids?
How the amino acids are getting combined.
Correct. So how the amino acids are getting appended? Correct. By getting
Mm.
if amino acids are getting appended, and what are the ways it can also even break?
So the ways
Correct.
that they can be combined and break.
Yeah. So amino acids can combine, amino acids can break. Fine. So amino acids also can form different structures. Form different structures. So before that, I'll give you a quick overview of what DNA too. Why do we need to know about the DNA?
Because that is what is making up our genetic.
Exactly. You're so correct. Right? So the
Mm.
fingerprints in your gene. Fingerprints is in your gene, correct?
Yes.
Yes. So the shape, what we acquire, the color of our skin, correct? Everything
Correct.
is based upon what?
DNA.
DNA. Fine. So do you know the difference between DNA and a gene?
Mm, DNA is what makes up a gene.
Okay. DNA makes up a gene. Then what about protein then? mRNA then? So these are the basics we want to get it revised. You're getting my
Okay.
point?
Mm.
Yes. So have you heard of the term called transcription, translation, replication?
Yeah, it's like using the adenine thymine in the DNA, right?
Correct. So to be simple, DNA will code into an RNA, and RNA will code into what? A
Um,
protein.
protein. Okay. So DNA to RNA and then RNA to
Yeah,
protein.
you can make a note of this. So DNA to RNA and RNA to protein. Correct?
Yes, correct.
Yes. So this DNA to RNA is basically called as what? Transcription. Transcription. So do you have the habit of watching Hollywood movies with the subtitle?
No,
Any
sir. Like,
complex? Yeah. So since you are in abroad, I think you are well versed in English. But what about Sherlock Holmes? Some complicated English stuff they may use. Correct?
I know about him, but I have not watched.
Fine. So here the content is what you have to understand is uh, watching an English movie with a subtitle is called transcript. You're
Transcript.
getting my
Yes.
you're getting my point.
Yes,
Watching a
correct.
English movie
And
with a subtitle is called as what?
transcript.
Transcript.
Transcription. Mm.
Correct. So DNA is. Can you please expand DNA? Quick check. Can you please expand DNA?
Expand DNA.
Yes. Full form of DNA. Can you expand the abbreviation?
Deoxyribonucleic acid.
Fantastic. Deoxyribonucleic acid. Fine. So now you can
Mm.
easily expand RNA too. What is the expansion of RNA?
Ribonucleic acid.
Correct. Ribonucleic acid. So now there is an answer hidden in that. Both are nucleic acids. Have you noted that?
Yes.
Both are what?
Nucleic acids.
Nucleic acids. So an English movie playing in English have the same subtitle in English. You're getting my point?
Yeah. Yes. Correct.
Correct. So that is called as what?
Um. Translation.
Transcription. That's it. Whereas if you're watching a Hindi movie, maybe you're watching a Hindi
Hindi
movie, correct?
with English subtitle, then it's called as translation.
Correct. You're getting my point? So that is
Yes.
called as what?
Translation.
Exactly. Now, DNA converted into RNA, RNA is transcription, but RNA converting into a protein is called as what?
Translation.
Why?
Because RNA is it's like the Hindi movie and protein is the English movie. So we are
Correct.
watching it in English subtitle.
Exactly. So now you can guess the answer because DNA, RNA is made up of nucleic acid, DNA is made up of nucleic acid, whereas protein is made up of
Amino acid.
fantastic. You got it, Adi?
Yes.
So that's a whole idea here, right?
Yes. Correct.
So these are the basics we have to know about the DNA.
RNA
Fine.
protein.
RNA versus DNA. So can we have a quick check that we will see through today? Then we also have a quick check about DNA structures, Chargaff's rule. Correct. That is very, very important for our understanding.
Mm.
And yes. Moreover, in the second class, like by tomorrow, next class, we will see about CADD.
Computer aided drug design.
Correct. Computer aided drug design. And slowly we will move on to CAD. Like what are the tools we are using. So I'll get to know your configuration of your system later. Okay. And you're going to do an hands-on with me. You're going to do an hands-on with me, and I'll give you a lot of worksheets. You can give a practice. Fine. And your assignment will be noted. Like let me confirm with the tutor. Like Ms. Bhavani, do we have a Google Classroom for Mr. Adi, Junior Adi? I hope so. Yes.
Can you please come
Like,
again?
yeah. So do he have a Google Classroom?
No, I'm going to create it right now.
Yes, correct. So so near future you will be having a Google Classroom, okay? Where you have to submit your assignments.
My doubt is regarding the assignment, or do you want to give him any documentary?
Regarding document, we can post in the Drive, Google Drive, and he have to submit the assignment.
Fine. Classroom will be created in a while. I will create it.
Sure, sure. Thank you. So Adi, what you have to do before the next class, you have to complete the assignment and post it there.
So what assignment, like worksheet?
Yes. You get. It will be differing. For example, I will ask you to browse something, but the first assignment will be with your hands. Like you have to make hands-on diagram. Don't take printout from anywhere. Take a pen and paper. Draw the structures. If you want, you can use color sketch. So that assignment will be quite straightforward. That is none other than a structure of amino acids.
Oh, okay.
Structure of
amino acid.
Yeah, you can make a note of it, and you will be getting a message also. Okay, the assignment will be structure of amino acids.
What amino acids should draw, like
all 20 amino acids with a classification.
Okay.
With a classification, and don't worry, we will have a quick go through today itself. Right? What are the 20 amino acids? I'll teach you in the class, but along with that, what you're going to practice? You're going to practice that in your home.
So I should like go to Google and draw it.
Yeah, you can Google to Google. You can find the images anywhere. Rather that, I will suggest you a better what? I'll suggest you a better book. I'll share you the book also in the Drive. Okay, this book is followed in the Google Classrooms. Like this book is followed universally. You're getting my point?
Yes.
That book I'll share with you. So that book you can later prepare.
Okay.
Fine, Adi?
Yes.
Yeah. So that we will see, and we also will see about different types of interactions. Different types of what?
Interaction.
Interactions. So if you remember somewhat in the last class, we discussed about covalent, non-covalent interactions, and you might have also studied in your classes, schools.
Yeah.
Can you just recall what are the types of covalent, non-covalent? What is the difference? What is covalent interactions?
So a covalent interaction is when two non-metal will share the atom, but an ionic electron, but in the ionic bond, two one metal, one non-metal will be sharing electron.
Okay, on metal, non-metal. So it's a kind of electron donor, electron acceptor concept.
Yeah, like if one is having 7 and the other is having 2, to have full shell, it will take the one from the 2 to give 8.
Can I name that as electrovalent bond?
Tetravalent bond and all they did not take, sir.
Fine. So Adi, the thing is, whatever you said is okay. Whatever you said is, it's called as um, it's called as non-covalent bond. Okay, where electron donation and electron acceptance are happening. Getting my point,
Hmm.
Adi?
Yeah.
Whereas what I'm going to discuss you today is covalent bond, mutual sharing of electrons. Mutual
Okay.
sharing of electrons.
Electron.
Fine. Yes. So the slides will be shared with you. So don't worry. You can see the recording too. Fine.
Yes.
Generally, we won't have the habit of sharing slides, but since you are a beginner, okay, I'm happy to share my slides. Keep it safe. Don't share with anyone else. Fine.
Mm. Mm.
And moreover, you also can watch the recording later.
Yeah.
Fine.
Okay. Makes sense.
Sure. Yeah. Okay, Adik. Right. So regarding other modules, I'll have a quick go through like Linux. So what kind of OS you are using now?
Mac OS.
Fine. So is there any possibility to have Linux side by side, like have a OS along with it? You can get the help of your dad or mom. Fine.
I can download it, like if you want me to.
Yeah, it's not a downloadable thing. Like you have to alter it. Like either you have to have dual OS or you
I
can have it's a completely new operating system. Okay, like you take the help of your parents. Okay, it's not now. I'm saying in the future classes.
Okay. Yeah.
Got it, Adi?
Hmm.
Yes, in future classes you also can download the
Linux.
OS and keep it ready.
Okay.
Fine. So yeah, then after that, okay,
Mm.
literature review. What is literature review?
Mm.
What is literature we
Like
call?
looking at the reading or something like.
Yes. So it's definitely a reading, but reading in our research term is called research articles.
Okay.
Before you begin any research, you want to know about what are the previous researchers have contributed in that.
Okay.
Fine.
Yes.
For example, you want to know about the aeroplane. You want to design an aeroplane. So where do you start with?
What contributions have been made to aeroplane first?
Yeah, correct. It starts with a very ancient time of making wheel.
Mm.
Am I making sense?
Yeah. So from
You're
wheel
getting
we
my point?
to
Uh,
airplane.
yeah, the wheel is made a very long back, right?
Yeah.
Even in the caveman time, caveman have prepared the wheel very long back. Am I
Yeah.
right?
Mm.
So there is no use of making a design of wheel again. So whatever the existing knowledge is there, that knowledge I'll carry forward.
Yeah.
Correct?
Mm.
So that's the whole idea here.
Okay. Makes sense. Yeah.
Fine. So that we will see. Then slowly we will move on to primary and secondary predictions. Okay. So this primary structure, secondary structure, I'll give you an overview today. Fine. So apart from that, modeling, docking, will be discussed later. Fine. So
Yes.
just a quick overview of the next five sessions I've given now. Let me move on to the session now. So today's session will be about fundamentals of biomolecules. Okay.
Yeah.
Is my slide visible, Mr. Julian Adi?
Yeah. DNA and Chargaff rule.
Yes. So let us start familiarizing with that. So officially, welcome to the day one. Okay.
Mm.
So myself, Dr. Chandrasekhar, where I completed my BTech, Masters, PhD, postdoc. You know IIT Madras?
Yeah.
Yeah, I'm an alumni over there. Okay. Now I'm working as an assistant professor research.
Mm.
Yeah. Right. So any point of your time you got some pus like this in your face? Like,
No,
have
sir.
you seen people with pus or pimples?
If oil come in the skin, it will come sometime.
Correct. Sebaceous gland. Correct. We call as what? Sebum, technically. Right?
Mm.
Okay. It's none other than the bacteria over there in the skin are getting counterattacked by our immune system. Got my point, Adi?
Yeah.
So what happens over there? You know about WBC, RBCs?
Yeah. White blood cell, red blood cell.
Fantastic. So white blood cells will cleave off, like the cells will be broken down. Got my
Mm.
point?
Yes, correct.
When the cells are getting broken down, what happens? It releases the. It
Mm.
releases what?
White blood cell. I don't know, sir.
Yeah, it releases none other than it releases lot of nucleic acids.
Okay.
So DNA, RNA of that WBCs will be released.
Yeah.
So so the scientist called Frederick Mischer in 1869, in the year of 1869, he have found that, okay, see, a cell consists of DNA, and the DNA he found from the pus of the infection. Okay, so that's a way. Research have started about nucleic acid. Again, it's a very interesting slide.
Mm.
Can you see some healthy mouse and a dead mouse?
Yes.
Right. Let me have a quick check. What is the difference between mouse and rat, Adi? What is rat? What is mouse?
Mm.
Have you seen both?
I've seen both, but they look very similar, sir. I don't know the difference.
Okay. The large one is called rat and the small one is called mouse.
Okay. Yeah. Okay.
I do not know whether you remember your mother tongue, like Perichali, Sundali.
Yeah. Yeah. Yes, I understand. In
Tamil
Tamil.
சொல்லுவாங்க Perichali, Sundali. Okay. So this is called a Sundali, a small mouse. Okay. Right.
Mm.
Let me go into the topic now. What happened? Another scientist called Griffith. A scientist called Griffith. Yeah, a scientist called Griffith. I'll share you the slides, Adi.
Okay.
No worries. I'll share you the slides. You can note only the important points, like what is not in the slide and what you feel very interesting. The code word alone you can make a note. Okay.
Oh, not in the slide. Okay. Yeah.
Yeah, which is not in the slide, that alone you can make a note. The
Mm.
code word. Okay. And again, I say, if you want to listen, you can go back to the recording and listen again.
Yes, sir. Okay.
So just focus. So what happened? He took a healthy, he took a bacteria. He took a bacteria with yes. Yes here refers to smooth. Yes here refers to what? Smooth. Smooth strain. Right? Then he injected to the mouse. What happened? The smooth strain is so pathogenic. So the mouse dies. The mouse was eliminated. Okay? Sacrificed. But what happened? He took another strain called R strain. See, here the strain refers to different forms of bacteria. Getting my point?
Yeah. So living R cell is like different bacteria.
Yeah. S cell is also a bacteria, a type of bacteria, and R cell is also type of bacteria. They are of same group.
Mm.
Okay.
Makes sense.
What is the only difference is their surface. Like,
So,
see, like we Indians also Homo sapiens, Chinese also Homo sapiens, Americans also Homo sapiens, but we look different, right?
Yeah.
Same way, the bacteria look different.
Mm.
So he tried with different types of bacteria. One is a smooth strain, S strain, another with the R strain. You get the difference?
Yeah.
What happened is when the smooth strain okay, is heat killed. This particular stain is heat killed. What happened? It loses its smoothness, and the mouse even survives. You getting my point?
Yeah.
So
So
they
that
are
is
giving
R cell and S
correct.
cell.
Yes. See, in simple, when I give yes to the mouse dies. When I give R to the mouse lives. The mouse survives. Clear?
Mm. Yes.
What's the third case?
When I give the heat killed S to the mouse.
Yeah. So when I heat kill, the virulence got reduced. The virulence got reduced and the mouse survives. Clear?
Yeah.
What happened in the fourth case?
If I mix the heat as heat killing S cell and living R cell, the mouse will die.
What is expected?
That it live.
Yeah, correct. No, because in this case also it's living. In this case also it's living. Why?
But if you
When
mix both, it's dying.
what happened, Adi, here?
If you're mixing both, it's dying.
What happened? What might be the reason?
Mm,
Yeah, the reason is given above. The thing is so simple. Okay,
mm.
the DNA is getting switched over.
Okay.
So this is the first evidence that DNA can jump from one cell to another cell.
Jump from okay. Yeah.
These are called jumping genes. DNA can transform from one cell to another cell.
Honest-
So I'm saying about the history of DNA discovery. I'm saying about the history of DNA
Yeah.
discovery. So in 1869, people have found the DNA in the pus. Okay.
Mm.
Later on, what they found, Griffith have found in the conducted an experiment, and he found that no, it is also can be found in the jumping genes.
Yes. Mm.
I don't want to go so depth in this, but these ideas you can keep in mind, Adi.
Yeah. 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 I know. Yeah.
Hershey Chase. Again, see the a question is followed. See, people are in surprise. Now we are having a very clear picture, but I'm saying some 100 years back, some 200 years back,
Okay.
or in roughly 150 years back. You're getting my point?
This makes
Today
sense.
is 2026. In 1869, people are not aware what is DNA, what happens from how you are getting your character of your mother and father, and
Father.
how your grandparents' characters also with you. Correct, Adi?
Yeah. So they did not know then.
They do not know that. They assume that okay, something is transferring, and later scientists have found that the transferring material is none other than DNA.
Okay. Yeah.
The transferring material is none other than
DNA.
DNA. So these again another kind of interesting experiment. This is called transduction experiment. I told you transduction. Do you remember?
You said transcription and translation.
Correct. Transcription and translation. This transduction is another thing called a virus attacking a bacteria.
Okay. Mm.
Virus attacking a
Bacteria.
correct. That is called transduction. Whereas this is called transformation. Okay. So you have
Okay.
to know about different types of these T's. Transcription, translation, transduction. Clear, Adi?
Oh.
Yes. Yeah. Who are these gentlemen? Any
Uh,
idea of them?
they won Nobel Prize.
Yeah, they won Nobel Prize. So at least you should be aware of Nobel. Am I right? Alfred Nobel.
Yeah.
So the
I,
biggest achievement in any scientist is none other than
Alfred Nobel.
acquiring Nobel Prize.
Okay.
So these scientists have given Nobel Prize for discovering the structure of DNA.
Okay. And that makes sense.
I see. We do not know how a DNA looks, Adi, correct?
Correct.
So later on, what happened? Scientists have found
How the DNA structure
how
should look.
how a DNA may look. How the DNA may
Look,
look. So how the DNA is looking here?
like it looking like a helix.
Fantastic. So that is found by X-ray crystallography.
X-ray diffraction.
Crystallography. So I know these terms are very tough for you, but you can assume. Have you ever seen some X-ray pictures?
Ah, yeah.
Any one of your relatives, your dad, mom?
No. When I got a X-ray for sprain,
Oh, oh my God! Now you're health. Now you're okay. You're doing
no, that
fine.
happened long time back, sir. Yeah, I'm fine.
Fine. So you yourself have experienced the X-ray picture, correct?
Yeah.
It's kind of similar X-ray picture of a crystal, small crystal.
Hmm.
See, we are a human body. They are passing X-ray in our body and they are trying to find. But here the X-ray they will just bombard on a small crystal.
Okay.
They take the DNA out of the cell and they will make a crystal. Okay, DNA is a very small thing. So they will accumulate millions of DNA together. On the DNA, they will bombard the X-ray.
Yes.
So from the diffraction pattern, see, X-ray is bombarding on it and they have a diffraction pattern, right? That
Yes.
diffraction pattern they have captured in the helical form. Are you getting my point?
Yeah.
So this is the overall story, discovery of DNA.
No, okay.
Like started, I told you about the Frederick Mitchell's thing, correct?
The Drake.
Yeah, these scientists you don't need to memorize, but just have a quick look. Like later they found that this DNA is made up of ATGC, 4 nucleotides.
Okay.
Later on, they found that this DNA is only having the genetic information. Clear?
Okay. Yeah.
So genetic information means what? Like a blueprint. Like
a blueprint of like what traits will be carried down, right? Like
exactly. For example, your mom is cooking some food.
mm.
If someone want to mimic the same, like your dad want to practice that food in your home, okay? So what mom have to share with dad?
How to make
A recipe. What
recipe.
amma have to share with appa?
Recipe.
Recipe, correct? So recipe means what have to be done after that. Okay, one after another. Same way, the recipe for your eyes, the recipe for your hair, the recipe for your skin is all from DNA. Right. So how many alphabets we have in English?
26.
Yeah, same 26 alphabets in different formats. How many words, how many sentences, correct? Different phrases, different sentences, different meanings. Same way, in biological terms, we have only 4 alphabets.
Okay.
What are the 4 alphabets, Adi? As you see in the screen.
A, G, C, T.
That's it. Simple.
Oh,
Say only 4. The combinations of these 4 are making the entire universe, you know?
Yeah.
All the living beings in the universe. Okay? So there is a famous Tirukural. Even that Tirukural is used by our current CM, Dr. Vijay, you know?
No.
Yeah, an actor Vijay. Okay, the Tirukural is Pirapokkum Ella Uyirkum. The thing is, all living beings are same. All living beings are
same.
Or in another terms, even from a bacteria, and you are same. The banana you ate in the morning, the banana and we humans are same. How come?
Because we both have the genetic information.
Yeah, everyone are made up of same DNA, but the combinations getting complex.
Yeah.
You're getting my point? So all living things in this world are made up of only the 4 alphabets.
A, G, C, T.
That's it. ATGC. We call in another way. ATGC.
ATGC.
Yeah, that's a simple. ATGC. So A pairs with T, G pairs with C. So these complex informations, see later on after this class, you should not stop. You have to go and browse through about the discovery of DNA.
Yeah, okay. I will look in Google.
Yeah, look into the documentaries. Look into some animation videos, how the DNA looks, and why DNA is important. Clear?
Yes.
Now you got this point, Adi?
Yeah, I can understand.
Very important point, correct? So this DNA carry the genetic information. Yeah. And another scientist in 1950 have found DNA is not a line, single line. DNA is a
base pairing.
Yeah, base pairing occurs. Base pairing means what? Double strand. Yeah, A pairs with T and G pairs with C. A pairs with T, G pairs with
T.
Okay, A pairs with T and G pairs with C. And again, there is a reason for these lines.
Is that equal to?
No. First is double bond, the second one is triple bond.
Oh, okay.
Double bond and
triple bond.
Triple bond. Triple hydrogen bonds and double hydrogen bonds.
Okay.
Clear? You can make a note of this double bond, triple bond stuff. So how many alphabets are there, Adi?
26. In normal, but in DNA it's 4.
Yeah, in DNA, the language of biology, it's 4. Okay.
Great. Yeah.
Yeah, ATGC. So this is the combinations of DNA. In RNA, instead of T, there will be U.
Uracil.
Yes, fantastic. Okay.
Mm.
And what pairs with what? A pairs with T and G pairs with?
C.
How many bonds are between A and T?
Two hydrogen bonds. GNC it's 3 hydrogen.
Fantastic. How many bonds in G and C?
3.
3. Superb. Got it?
Mm.
So these are the chemical structures of the okay. These are the
DNA.
chemical structures of the that 5 nucleotides.
Yeah.
So this RNA, this uracil occurs only in RNA,
RNA.
and thymine occurs only in
DNA.
yeah. Other 3 are common. Other three are called as what? Common in both DNA and RNA.
Yeah.
And again, if you know technically, they are called purines and pyrimidines.
Pyrimidine. Okay.
Yeah, pyrimidine. Fine. Purines and pyrimidines. Got it?
Yeah.
And a quick overthrow about these kinds of bonds. So, are you aware of this?
No, I don't know about like pentose.
Fine. So these are the carbohydrate moieties. You don't want to go deep into it, but at least have a quick look. So that, yeah, as the name suggests, Adi. So what's the name of DNA you told?
Deoxyribonucleic.
Fantastic. Deoxyribo, correct?
Yeah, ribo is there.
This is the ribo. This is the ribose.
Yeah.
Clear?
Mm.
This is the ribose, pentose. So they are
So-
having,
Bond is ribose. Okay.
yeah, and this base is the right hand side wala.
Okay. Yeah.
So this is the nitrogenous base. This 5
Okay.
steps are nitrogenous base. So these nitrogenous base are bounded with ribose,
Okay. Yeah.
and they form nucleoside. They form what?
Nucleoside. Okay, not
Yeah.
nucleotide.
When the nucleoside combines with phosphate, they form,
Nucleoside monophosphate.
yes, or in simple terms, they can be nucleotide. Simple.
So nucleoside monophosphate is nucleotide. That's it.
Yes. Yeah. Again, these nucleotides are generally having 3 phosphates.
Mono,
Like
bi, and tri.
yeah, the standard form is triphosphates. We call ATP. See, actually, adenosine triphosphate. Have you heard of this adenosine triphosphate somewhere?
Yeah, ATP energy.
And fantastic. These ATPs are the energy currencies of the cell. Clear?
Okay. Yeah.
So when it is in energetic form, they will be having 3 phosphates. When it is in a normal form, like in a DNA form, they have only nucleoside monophosphate. Clear?
Yes.
So these things you have to revise. Like you can make a quick notes in your notebook somewhere. Like
Like
nitrogen.
why you revise exactly? Like
Yeah, please. You revise it whenever you feel free. Like nitrogenous base plus pentose sugar is what? Nucleoside. And nucleoside plus a phosphate is called as what?
nucleoside monophosphate.
Nucleotide.
Oh,
What is
okay.
a quick way to call it?
Nucleotide. Yeah, sir. I took a screenshot of this. I'll look at it.
Please, you take. And I'll share the slides too. Okay.
Okay. Yeah. I screenshot it, so it's fine.
Good. Fantastic. Yeah. Now a quick check about DNA and RNA. So DNA is double-stranded. RNA is
single-stranded.
fantastic. So what is the full form of DNA? I told deoxyribonucleic acid.
Ribonucleic acid. This is just ribonucleic acid.
Can you look at the quick difference between the two structures of ribose? With the clue of this word?
Yeah. So then, so there is a two oxygen hydrogen bond. Like there should be two.
Fantastic, man. You got it. This one, right?
Yeah. Yes, that one. In DNA it's H, and in RNA it's OH.
Correct. So this oxygen is missing here.
So
Actually,
ribose is having extra oxygen in.
yes, that is the actual structure of ribose. Ribose is the actual structure. When they form DNA, it will lose its oxygen
Oh,
to
okay.
maintain stability. Okay,
Yeah.
this is again a big story. Okay. And another important thing, DNA will be preserved. DNA cannot transform into a protein. DNA
But
have to, yes, DNA have to transform into an intermediate called RNA. Then RNA will transform into a protein.
Mm.
If you know the entire story, DNA will be formed inside the nucleus. Okay, DNA will convert into RNA into nucleus. This RNA have to come out of the nucleus. Clear?
So after they exit the nucleus, it will become protein.
Yeah, with the help of other stuff called ribosomes. So yes,
Right.
your cell biology classes go over. Like in your school, have you completed the cell biology courses?
Yeah, I
Like,
think so.
are you aware of mitochondria,
Yeah,
endoplasmic
mitochondria like
reticulum?
powers of the cell. Yeah, I think I learned.
Fantastic. What about the endoplasmic reticulum, ribosomes?
Isn't that where ribosome is found?
Yeah, correct. Again, in ribosomes, there is a peculiar ribosomes called rough endoplasmic reticulum.
Oh, yeah, that I did not learn.
Yeah, in rough endoplasmic reticulum. Okay, later on you have to listen to this audio, this video, and you have to go and browse it.
Yeah,
Okay, Adi? Because in your young age, you are planning for a big dream, correct?
yeah.
When you want to achieve the big dream, you have to work hard.
Mm.
And I understand that you are a very enthusiastic kid. So you are very interested to learn stuff. I can understand. Okay,
Mm.
you can go and watch animations. Lot of animations are there. Okay, if you want,
Okay.
I can share the link too. Right? And you
No,
want to understand.
I'll go to
Yeah,
YouTube and I'll find it.
yes, please. So you want to know the difference between all these
Um,
steps.
the cell parts.
Yeah, cell parts. How the transcription occurs, how the translation occurs.
Mm.
Right? Then only you can understand the importance of DNA and RNA. Clear,
Yes.
Adi?
Makes sense.
Yes. Now this is Chargaff's rule. The beginning of slide, I told you now what
Yeah,
is
I'll
Chargaff.
write this.
And you remember, I just drawn this bond. A double bond T and G triple bond.
See.
Can you see the bonds over here?
Yeah.
It's 1 and 2. It's 1, 2, 3.
Okay.
Clear?
Yeah.
Yeah. This slide says about how many A is that, that many T will be there.
Okay.
How many G is that, that many C will be there. But this is not right. A plus T is not equal to G plus C.
So A plus T is not equal to G plus C.
Correct.
Okay.
The reason is, see, when I hold you with 3 hands, okay, so okay, you have 2 hands, right? Left hand, right hand.
Yeah.
So Appa is holding you with 3 fingers, and Amma is holding you with 2 fingers. So which holding will be very strong?
Done with 3 fingers.
Yeah, same thing, correct. So yeah, some part of the DNA will be very strong, tightly
Yes.
bound. Some part will be loosely bound.
Okay.
Okay? So that all depends. It not necessarily to be same. Clear? Again, I don't want to go into a deep sketch, but in short note, you can understand it's a helical structure,
Helical.
and they are in the size of 0.394 nanometer. So DNA is in the form of,
0.34 nanometer.
yeah, they have a distance. They are a very distances in nanometer.
Yeah.
And here, yeah, here I want you to imagine a small stuff. I keep a dot here, Adi. I keep a dot, and I
Where?
want you to, can you see a dot over here?
No. Oh yeah,
Here?
there. Yeah. Okay.
Okay, this dot you try to cut into 1,000 pieces. Is it possible?
It's possible but difficult.
You can't imagine, right?
Yeah, it'll be very small.
Very, very small, right?
Mm.
We can't even see by eyes. Am I right? Okay. That unimaginable dot, you're going to imagine now. That unimaginable dot, you're again going to cut into 1,000 pieces.
Oh, that is also even very, very smaller.
No one can see by naked eyes, correct?
Yes, not possible.
Yeah, let me try to figure out this analogy. Whatever the dot you are seeing is 1 millimeter. Whatever the dot you see is
1 millimeter. Okay.
Now, when I cut that 1 millimeter by 1000, so what is a meter now?
Is it
Can you
nanometer?
write? Okay, no. Can you write this in power?
1 to the power of minus.
No, I should not say
Oh,
in
sorry.
1.
Oh,
10 to
yeah,
the
it's scientific notation. Okay. 10 to the power of minus 3.
yes. So how many zeros were there? That many zeros you
Yeah,
have to write.
yeah, that's all I mean. Yeah.
So 10 power minus 3
3.
meter. This is called millimeter. Okay, Adi?
Okay.
When I cut this 10.3 millimeter by 1000, then what it is called?
Nanometer?
No. This 10 power, this become 10 power 3. Am I right? 10 power
Yeah.
minus 3 by 10 power 3. Correct?
Yes.
When it goes above, it becomes again
Positive, right? Like,
negative. The below one is positive. You are getting my point?
yeah, this is unclear. I can't see.
You can see?
Yeah.
It's a new slide. See.
I mean,
10.
I
Can,
can see that one. Yeah.
can you see 10 now?
Yeah, now I can see. I enlarged my screen.
Yeah. 10 minus 3, can you see?
Yeah.
Into 10 over 10 power minus 3, what it becomes?
10 power 0, which is 1.
10 power minus 6.
No, 10 power 3 into 10 power minus 3 is 10 power 0,
So
right?
10 power minus 3 into 10 power minus 3.
Yeah, then 10 power minus 6, yeah.
Correct.
Hmm.
This becomes your micrometer. This becomes your what?
Micrometer.
So this is the dot I told you. Take a dot, cut into 1000 pieces I told, right?
Hmm.
How small the dot will be. That we made a mathematical measurement that is called 10 power minus 6. You got it now?
Minus 6 nanometer, okay.
Yes. This is called as micrometer. This is called as what?
Oh, micrometer. Hmm.
This micrometer is the size of all microbes.
Microbe means?
Microbes means microorganisms, bacteria, fungus,
Oh, yeah, okay.
even our cells. Even our cells are, you are saying neuron, right? Neuron,
Yeah,
your
yeah.
lung cells, liver cells, all blood cells. All cells are in this small shape, size.
Yes. Hmm.
I'm trying to help. I'm helping you to imagine the size of nucleus cell.
Hmm.
Now coming back to the story, I told you to cut 1,000th piece. Now that 1,000th piece you cut into another 1,000.
So 10 to the power of minus 6 divided by 1000.
Yeah, what happens again?
That 1000 will go to the numerator, becoming 10 to the power of minus 3. So 10 to the power of minus 9.
Fantastic. So do you know that term of 10 to the power of minus 9?
Scientific notation.
Yeah, what? Nanometer.
Oh, nanometer. Okay.
Clear?
Yeah.
Yeah, this and all you have to learn. Okay. What is
Mm.
millimeter, micrometer, nanometer?
And until millimeter, I know. After that, I don't know.
Yeah, you have to learn. Okay, Adi.
Yeah.
I'm happy that you know it already, like till millimeter. Other things you have to learn. That
Yeah.
nanometer is the size of the DNA. The nanometer is the size of.
DNA.
Can you see here that how small the DNA it is?
Yeah, very, very small.
Very, very small. We can see only by electron microscope. We can't see by our normal microscope also.
Okay. Yeah.
Yeah. So there is a crime scene. Okay. Imagine there is a crime scene. Okay.
Okay.
In initial days, in the grandfather's time, if they want to find the crime, they will look at the fingerprints of the culprit.
Okay.
Have you seen some Hollywood movies or Indian movies where they detect the crime scene?
Do this to the place and they'll find out the fingerprint.
Yeah. But what happens in real time, you know, they try to find the crime scene. They won't look at the fingerprints nowadays. They
Okay.
look for samples, either blood stains. Okay. They will look even for hair follicles,
Yeah. Okay.
and they bring to lab and they try to detect the DNA of the culprit.
Yeah. Okay.
Culprit plus suspect. See, they collect the DNA. See, they got a hair follicle from a crime scene. Okay. But with that hair, I can't find whose hair it is. Can I go and check each one's head?
No. That will be long time.
That is not possible, technically possible. What if the suspect go and color his hair or her hair?
Yeah, that makes sense.
Yeah, rather what I can do, I can find the DNA in that hair.
Okay. Yeah.
I will find the DNA in his hair. Then what I'll do? I'll
I
just
know.
doubt. Yeah, I'll just doubt 4 to 5 people using some clues. I have found that using CCTV camera or something. I found some 4 to 5 people.
Then you'll find that, and then you check their DNA and see how it compares.
Ah, correct. Look at the below picture.
Okay. Yeah, I'm seeing.
Can you see the below picture?
I can see it.
Yes. Where I got. Okay. Okay. Over here. Over here. Can you see?
Yes.
So see crime scene. Whatever the DNA we got in crime scene is with suspect B.
Okay.
See, by naked eyes, I can see this guy is looking rugged. So we should not judge him as a culprit.
Yeah.
Rather, this clean shaved guy might be the villain.
Okay.
So who helped me to find the answer?
The
DNA.
DNA machine, right? Like that.
Yeah, the DNA machine is called PCR machine. Okay.
I think thermal cycler.
Yeah,
Okay.
this one, this one. This is called PCR. And this
PCR.
you can study in your future classes like polymerase chain reaction.
Mm.
I don't want to go so deep, but these slides you can go and check.
I'll see after the class.
Sure, 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 sure. It will be the slides will be shared in the Google Drive. Okay, Adi.
Yes.
Yeah. So look at this. Okay.
Mm.
Yeah, this is the difference between DNA and gene.
So gene is just a small section, while DNA is the entire thing.
Correct. See, if I want to write, I am saying that I'm claiming that I'm writing with my hand. Do I write with my whole hand?
No, you only take like 3 finger and write.
Correct. So now the whole hand is DNA and the 3 fingers is a gene. Got an
Oh,
answer?
yeah, now I understand.
The reason is, then you may say, sir, I can have only the 3 fingers, right? Why should I have the lengthy hand? So hand is also important. The fingers also important. Clear?
Yeah, different role it will play. Yeah.
Yeah. The hand is a structural part. Fingers are the functional part.
Okay. Yes.
Got it, Adi?
Mm.
The functional part is this small fingers having information for coding into a protein. Clear?
Yes.
Now you got what is protein now?
I can somewhat understand.
Very simple. I told you blueprint, right? You all remember blueprint?
The gene blueprint.
Yes. See, I told, see, for example, see, you are a very kid man. How can I make you understand? Do you know about sperm, ovum?
Yeah, I know. That.
Have you ever heard of sperm, ovum?
Yeah, it's like the male reproductive and female reproductive.
Interesting. So, sperm and ovum will carry the information of your entire heredity.
Oh, okay.
See, from what you got from your dad, sperm or ovum?
Sperm.
Fantastic. So that sperm is having the information of the entire father's history.
Mm.
Same, take it scientifically. Same way, your the mother's ovum is having the history of your maternal history. You're getting my point. So
Okay.
what happens here is that information will be stored in the form of only the 4 alphabets. What are the 4 alphabets I told Adi?
ATCG.
That's it. This ATGC 4 alphabets now it's going to transform into 20 alphabets.
Mm. Mm. Okay.
Looking complex. The story is getting heated up now.
Very
Okay,
deep.
no need to get panic. This is called molecular biology. Okay.
Mm.
In simple, I'm trying to make you very
Okay,
clear.
so just sorry to interrupt you. I think you are discussing something quite interesting, but sorry to interrupt you. Yeah, as per schedule, it's planned for an hour, and we have 5 more minutes to complete the session. I just want to remind you this.
Oh, okay. Thank you, Ms. Bhavani. Can I have 10 more minutes, please?
Yes, sure.
Thank you so much.
Yes.
Yeah. Yes. So, Adi, you got the point here?
Yeah. Very complex.
Very complex, but don't confuse. This is just a combination. This is just a number game.
Okay.
Just a number game. For understanding this, you want to know some basics of mathematics.
Oh,
You
like
know,
in jeans we will have Punnett square, right? Like that. It's looking
yeah, like Punnett square. This is called as genetic code. This is called as
genetic
what?
genetic code.
Genetic code.
Yeah. Okay.
Very simple. Look at here. So let me write your name. Your name is visible.
Sir, I don't think you're sharing screen.
It's still shared, man. Like it's not. Stop sharing.
Yeah. I just see you. That's it.
How about now?
Um. No, not yet, sir.
Just a minute, Adi, please. Okay, a small task for you. So I have your 4 letters, Adi, correct?
Adi. Yeah.
Can you see your name?
Yeah. You put here. Yeah.
Yeah, what are the different combinations? I want to make only three letters of your name with
So
different combinations.
I have 3 spots, and then I am having a 4 letter where order doesn't matter. Yeah.
Yeah, order doesn't matter. So how many combinations you can make, Adi?
NCR.
Do you want to give a try in S class? Within S class?
Yeah, I can do it. Now, so I'm having. You want combination, right? Not permutation.
Yeah, how many combinations can be possible? You can make a
Uh,
quick answer. Like ADH, DHI, IHT, DHA, correct?
mm, four. Four
Only four
combination.
combinations. No possible.
Because permutation. Because if you want like a permutation is different in combination.
Okay. Uh, okay. Permutations. Make it permutations.
24 then.
Only 24. I said
Four
triplets.
because there is only 3 spaces and we're having 4 letters. So 4, then 1 letter is gone, then 3, then 2.
Okay. Actually, if you make actual combinations, you will get at the minimum 64.
How, sir? Can you explain like
Sure.
mathematically?
Sure. Okay. I told you triplets, correct?
Mm.
How many possibilities are there?
3.
Yeah, 3 possibilities are there, correct?
Mm.
Technically, it is 4 into 4 into 4.
But sir, if I'm having the 4 letter, then the next space only 3 will come, right? Because 1 letter is already used.
Nothing wrong. You can reuse.
Oh, I can reuse then. Yeah, 4 into 4 into 4 is correct.
Correct. That's what
Mm.
I told. Not only you should not restrict. Like I'm not saying you to restrict.
Okay.
You can reuse even.
Mm.
Okay. You're getting my point?
Yeah.
Reuse in the sense, see, AH, if I'm making ADH, the next combination I can use even, I can write in reverse way. HDA also another combination. You're getting my point?
Yeah, makes sense.
So by that way, how many combinations are possible?
64.
64 combinations are possible.
4 power 3, 64.
Correct, 4 power 3. I want you to make a table with your name in the next class. Take that is also homework. Okay, just for the fun. But main homework is what? The 64 will have some pattern of making 20 amino acids. How many amino acids?
20.
20 amino acids. Since we are running out of time, I will just quickly show it. Okay, you go through. The next class, I'll try to explain you.
Okay.
Clear?
Mm.
So these parts you just ignore now. I'll explain. Okay, I'll share only what is required now.
Okay.
This part I will explain you later. I'll send you the slide. I'll upload the slide in the drive. This whatever. Can you see this?
Yes, I can see. Non-polar. Okay, I just screenshot this.
Yeah. I'll share you the slide too. You can take a screenshot too.
I already took.
Yeah. This is the homework for you, Adi.
Okay.
See this DNA. You're getting my point?
Yeah.
Same like Adi into 4, 4, 4, and 4 into 4 into 4. Same way, 4 letters. Do you remember AUGC? I told you now.
Yeah.
Instead of T, it will be U. Because it's RNA, right?
Okay. Yeah.
Only RNA can transform into protein, not a DNA, correct?
Yes. Okay.
Got it?
Hmm.
So how many combinations are possible? Same like Adi. How many combinations are possible?
A 4 into 4 into 4 into 4, right?
Yeah, 3 times. 4 into 4 into 4.
Because there's only 3 spaces available.
Yes. So 64 combinations, same like your name.
Okay.
So in the 64 combinations, 61 combinations will code for different amino acids.
Okay.
For example, U will code for an amino acid called phenylalanine.
Phenylalanine. Okay.
Same way, A will code for
The
leucine.
Can you show where is sin coming from?
See, look at here.
Yeah.
The third letter is written here. A.
Okay.
Look at the chart. I think my point. A.
Mm.
Same way. Look at what are the combination. CGU. C.
E, G, and then U. So
G.
R.
U. So they will code for amino acid called arginine.
So iron will come at the end.
Yes. So what I'm trying to say,
Iron.
arginine, not iron. Arginine. Name is arginine. Okay. So this,
Yes.
look at this. I'm going back to this. So what are the critical terms you have to learn before we go into the project is, I want you to get familiarized with all these names.
All the names. Okay. Yeah.
With the structures. Until you are not clear with this, we can't do much research on this, Adi.
So non-polar, it's like valine and everything. I should look at it. Okay.
Yeah, the entire thing. You want to know what are the combinations. You want to know about the 3-letter code and 1-letter code.
Okay. Yeah.
3-letter code and
1-letter code.
See, arginine can be otherwise called as ARG, and they also can be called as R, capital R. Clear?
Yes.
Same way, histidine can be called as what?
H.
R otherwise, that is 1-letter code. What is the 3-letter code?
Where is histidine?
Here.
HIS or H?
Correct. Same way, look at tryptophan.
TRP or W?
Fantastic. Same way, next class, you want to say without seeing it. You have to acquire in such a way.
So we say protein, I should say PRO, like that.
Yeah, because in future, I'm going to give you a lot of softwares where you are going to visualize the protein. You want to be very thorough with the 1-letter code. Clear?
Yeah, okay.
Got it, Adi?
Yes.
So any doubt in it?
No, sir, I understand. I'll do this.
Yes. So these stuffs we have to make. So let us take last 2 minutes for your discussion. You have any doubts in this?
No, sir. I think I understand everything, like about the different amino acid and Chargaff rule and all the scientists have contributed to DNA.
Correct. So I started today's class with where we started our
Frederick.
Yeah, where we started our DNA discovery. Correct. Discovery of DNA. Am I right?
Yes.
Then we have moved slowly into the
Chargaff
structure of
rule.
Chargaff's rule and structure of DNA. Correct.
Yeah.
Scientist discovery, structure of DNA. Then we slowly moved into how DNA will transform into amino acids. Correct.
And then Nobel people who are one, and then DNA structure.
Nobel laureates made it. And I also told you an example, the power of DNA. Correct.
Yes. Okay.
Yeah, using power of DNA. And this is an interesting stuff. You can later read about this DNA replication. Okay.
Yeah, okay.
Difference between gene and DNA. Correct.
Yes.
Now you can understand how each and every amino acid can code. This I will explain again also, but you can have a look.
Yeah.
For this, you want to know cell biology.
Cell biology.
Yeah, I told you, like what is DNA, what is RNA, what is Golgi bodies, what are ribosomes.
Yeah, I will also like look at that.
Yeah, you look at
Good.
that part. Clear?
Mm.
And what's your homework, Adi?
Look at the amino acid structure and also create a table of the name.
Table of the name and draw the structures.
Oh yeah, draw the structure. That structure homework is
Very, very
taken.
important. They have an amino group, they have a carboxyl group. Okay.
Yeah.
Amino group and carboxyl group and one hydrogen, and the remaining is an R group. Remaining is an
R group.
R group. R group is residual group. It differs in each amino acids. Can you see different amino acids? They're having different R group. All other things are common. Clear? Hope so, no doubt.
No, I understand. I took note in my notebook.
Super, Adi. So you can find the slide in the Google Drive shortly. Okay.
Yeah, okay, great.
So before we are ending the class, any quick doubts?
No, sir.
Okay. So thank you so much for your patient listening.
Thank you, sir. I really enjoyed the session.
Thank you so much, man. I really, it's a great honor for me for you getting a feedback from you like this. So, Miss Bhavani, like, you want to add up anything?
Yes, thank you, Chandru. Hadi, how was today's session?
Oh, I loved it, ma'am. It was very nice.
Okay, fine. Glad you loved the session. Fine. Can you please check your WhatsApp group?
Actually, Amma is having a phone now.
Okay, no problem. Kindly check once you get it. I have shared you the classroom and drive link. So I'm kindly requesting you to join the classroom using your Gmail account. Okay. Once you join, you will be seeing a session called recordings and study materials. In the study materials, I have already attached a drive link where you can access the materials that are going to be given by Dr. Chandru. Okay.
Okay.
In recordings, you will be receiving the recordings link. Today's recording will be available by tomorrow. Okay. Now it's morning for you, right? So it will be around next day morning mostly.
Yeah,
Fine?
great. It makes sense.
Yes. Yeah, that's it from our side. And also you will be receiving the feedback link in the group. You can feel free to post your feedback. If you have any constructive things to take forward, you can even post that in the feedback. Okay.
Okay.
Thank you, Adi, and thank you, Dr. Chandru. And adding the next session, we will be updating in the WhatsApp group. Okay.
Okay. Thank you, ma'am.
I think you can note all this point and you can check it out once you get your mobile. Fine. Thank you.
Bye.
Thank you. Bye.